All right, thank you so much for that introduction. I've had a wonderful past couple of days getting to brainstorm already with many of you about different tool ideas and different experimental ideas. And I'm excited to share just one story of what I've been working on during my own transition phase in the Ting lab. And so in line with this year's showcase, so we're motivated by this general problem of this general very hard problem of discovering diversity in the brain. And when I think of diversity in terms of two different types. So one in terms of functional activity, so heterogeneous activity of individual neurons in different brain regions across the mammalian brain in this case. And so understanding which brain regions are involved in particular behavior or processing some sensory input. And also what are the specific patterns of neural activity or spiking that encode or predict that behavior. On the other hand, you can think about diversity in terms of gene expression. So again, we've heard this question posed again and again, what defines a cell type? And also one thing I'm interested in is what regions across the brains have shared cell types. And so this question has been posed, what do we actually gain from incorporating both firing dynamics and transcriptional and cell type constraints into existing models of brain function? So in order to begin to even get to this question, I think we really need to think about technologies that allow us to simultaneously access functional activity during behavior, so in the wake behaving animal, and the transcriptome of these cells. So over the past decade, and really during the whole time that I've been studying neuroscience, there's been this incredible, amazing explosion in tools that allow us to record now up to thousands of neurons from distributed across the mammalian brain using things like neuropixels, which we've heard talked about during this showcase. And also on the molecular readout, we now are creating these really large cell atlases of the entire transcriptome of the CNS and the peripheral nervous system as well in these organisms. And so now the challenge is how do we merge these technologies? And I think there are two general approaches that are being taken right now given the existing tools that we have. And so both are challenging. So the first is you can imagine doing real-time neural activity recording, so imaging-based neural activity using voltage or calcium indicators. So here you have access to these fast neural spiking dynamics that you want to see during a behavior. And then following your sort of recording of interest, you can perform in situ sequencing, and then you have a very hard image registration problem to sort of align this information back to your neural activity dynamics. So the second approach, which is the approach that we're taking currently in the Ting lab, is this idea of molecular neural activity recording. And so this is when, rather than looking at these fast spiking dynamics, you can imagine integrating the number of spikes that occur during your time window of recording. And just the neurons that exhibit these high amounts of neural activity spiking, you turn on a transcriptional reporter in just these cells. So you're recording a sort of history of the activity of the neuron in the actual transcriptome. And then you can read that information out using, again, in situ sequencing, or in this case, single-cell RNA sequencing. And so, again, there are trade-offs between these two different approaches. And one thing I like about this general approach is that I think, in the immediate future at least, it's much more scalable across the entire brain and in multiple deep brain regions at the same time. All right, so when you're designing a tool to create one of these molecular neural activity recorders, what are some of the considerations you want your tool to achieve? So the first is, how do you set the time window of recording? So how do you indicate to the tool, like, this is when I want you to be recording neural activity? And then the second is, how do you detect this neural activity that's happening? And this is the sort of signature that you're going to turn into a transcriptional readout. So again, there are two you can classify these types of tools into two groups of what we have right now. So the first is immediate early gene-based tools. So these are what the FOSS-TET tag or the TRAP2 mouse, for example. So in this case, you have an immediate early gene promoter, so CFOS or ARC. And this drives expression of a transcription factor or a Cre recombinase. And so to gate the time window of when that CFOS promoter is active, you can inject a drug in the animal, such as tamoxifen or doxycycline. And so these are the two sort of inputs into this AND gate logic. So you have to have immediate early gene expression, which is the reporter of neural activity, and you have to inject this drug into the animal, which gates the time window of recording. And so this other class of tool, which is much more recent, and one of the first examples was developed in Alice's lab earlier a couple years ago by Wenjing Wang. And this is a tool called FLARE. And this is a calcium-based transcriptional reporter of neural activity. So in this case, rather than immediate early gene expression, we're relying on intracellular calcium dynamics in the neuron to report the neural activity. And then to gate the time window, we're using blue light instead of a drug. So what this enables us to do is have much higher temporal resolution for our recording. So rather than the hours it takes for the drug to be metabolized in the animal, we now just have light that we can deliver and turn on and off at will. And we also believe we have higher specificity, as intracellular calcium we think is a much more sort of universal readout of neural activity than immediate early gene expression. All right, so what are the basic components of this FLARE tool? So we have three components. The first is the reporter gene that we express inside of the cells. So for the purposes of the talk, mostly we'll be using just a simple fluorophore. But really, this is a modular system. Whatever protein you want to express as your reporter, you can use. To activate this transcriptional reporter, we use a transcription factor, TTA. And this TTA is normally tethered to the cell membrane outside of the nucleus. And it's tethered to the membrane via a protease cleavage site, which is fused and protected by a blue light sensitive protein called the LOV domain. And then this little complex is tethered to the membrane via the calmodulin sensing protein MK2. So to actually release this transcription factor so that it can enter the nucleus in a calcium-dependent manner, the third component is this protease that we express in the cell. So this is an orthogonal protease to mammalian cells. It recognizes its specific protease cleavage site and doesn't cleave any other endogenous proteins inside of the cell. This protease is fused to the calcium sensing protein calmodulin. So these are the same two calcium partners that we all know from GCAM, calmodulin and MK2. And so what happens in our cell that now expresses this molecular recorder is that when there's high calcium, this calmodulin and MK2 interact and bring that protease close enough so that it's able to cut its cleavage site. And then the LOV domain in the dark of the neuron cages that cleavage site so that that protease is unable to, even if it's nearby, it's unable to cut it unless you deliver blue light to activate the LOV domain. So you have to have these two inputs inside of the cell in order to get a readout of high neural activity. So you have to have the blue light to activate the LOV domain, and you have to have the calcium to bring that protease nearby to cleave the TTA. All right, so that was the first generation of FLARE, and then a postdoc in the lab, Mateo Sanchez-Lopez, used directed evolution of the TEV protease in yeast cells to generate a much higher catalytically efficient protease that we've now incorporated into our next generation of FLARE, along with some rational engineering of the LOV domain to create a tighter LOV domain. And so this is how this tool works when we express it in cultured neurons. So in the dark, when there's no light delivered to the system, even if we're delivering electric field stimulation to these neurons for 10 minutes, we see very little mCherry reporter expression in these neurons. But as we deliver this electric field stimulation and light for 30 seconds, a minute, 2.5 to 10 minutes, you can see an increasing number of mCherry positive cells that we're able to detect. And we're really excited about this because the first generation that we were looking for required, at a minimum 10 minutes in order to even begin to see any TRE-mCherry activation. So we've gone from 10 minutes down to 30 seconds. So this is in cultured neurons. We want to make sure we maintain this high efficiency in vivo. So to test the sensitivity of FLARE in vivo, we took advantage of another blue light gated tool that I know well from my graduate studies, so the channel rhodopsins, these opsins ion channels. So what we did here is in a CHR2 transgenic animal, in the prefrontal cortex, I injected the FLARE viruses and implanted an optical fiber. So in this case, when I deliver blue light into the brain, two things are happening. I'm setting the time window of recording for FLARE, and I'm also activating these neurons that are expressing CHR2. And so immediately following the time window of recording, I'll begin to get transcription of my mCherry reporter. And then about 18 hours later, I'll have enough protein to actually visualize through immunofluorescence. And in contrast, a control animal, a wild-type animal that's still injected with the FLARE components, you might have some spontaneous neural activity during this FLARE recording when I'm still delivering the blue light, but it won't be enough to drive high amounts of this reporter, this TRE-mCherry reporter. And so this is what the data look like. So these are in these transgenic animals that express CHR2 and FLARE. And so in the mice that have the opsin, we see many more mCherry positive cells in the prefrontal cortex compared to CHR2 or wild type, CHR2 negative wild type mice. So this was first just during 10 minutes of optogenic stimulation, and then we kind of narrowed this down to even just delivering 1 minute of blue light pulses into the brain. And even in this case, we're able to see, detect more mCherry in these CHR2 positive mice. And this is just showing the protease that's fused to a BFP so we can be sure that we're actually expressing our FLARE recorder in the brain. All right, so we can already sort of imagine, very exciting experiments that we could perform now with this new and improved FLARE to study things, to, sort of now gain genetic access to ensembles of neurons during, very short time windows of behavior or sensory stimulus presented to the animal. But one thing I was interested in trying to use this technology for was discovering something about cell type diversity in the brain. And for this, I wanted to apply this to study a group of neurons in the nucleus accumbens that I was studying during my PhD when I was a little bit more focused on systems and circuits neuroscience. So the neurons in the nucleus accumbens are both functionally and molecularly diverse. We have these at least two main subtypes, the direct and indirect D1, D2 neurons. And importantly, they receive multiple different types of inputs, long-range inputs from throughout the brain. And so a question is, can these nucleus accumbens cell types sort of be further subdivided into new cell types based on what kinds of inputs they receive? So to imagine using FLARE to answer this question in vivo, so in an intact behaving animal, you can imagine injecting FLARE into these downstream nucleus accumbens neurons, and then you express CHR2 in genetically defined upstream long-range inputs. So when we deliver blue light now into the accumbens, we're both stimulating these upstream axon terminals and also with FLARE labeling any activated downstream nucleus accumbens cell bodies. So in essence, we're able to perform anterograde functional activity mapping using this technology. All right, so the specific input that I studied in grad school was these glutamate axon inputs into the nucleus accumbens. And my research, and also now many other people have shown that glutamate inputs into a specific subregion of the nucleus accumbens can signal aversive outcomes to the animal, so it can drive avoidance behaviors in these mice. And the question is whether these glutamate axons are accessing a particular subtype of nucleus accumbens neurons, and whether reactivation of those specific nucleus accumbens neurons can then cause these same aversive behaviors in animals. All right, so first we just need to confirm that these accumbens neurons actually spike calcium spikes in response to glutamate inputs, so they don't fire very often. We want to make sure that this is a very robust input that drives neural activity. So this is just some green lens imaging of the nucleus accumbens of neurons expressing a red calcium indicator and that have the CHR2 expressed in glutamate axons. And so this is one photon data, but once we perform the CNMFE background subtraction, what we can see is that when we sort of sneak in these blue light pulses in between imaging frames, you have some subset of neurons that exhibit reliable responses, so calcium spikes to this axonal stimulation, others that maybe spike some of the time, and then other neurons that are active but don't seem to reliably respond to the blue light stimulation. So the question is, how do we genetically target and understand more about these neurons up here? All right, so this is the general idea of what we're doing. So injecting FLARE in the accumbens where you have these incoming glutamate axon fibers, deliver blue light to start the FLARE recording, and then in just the neurons that are functionally coupled to these glutamate inputs, we expect to see a tree reporter mCherry. And so the question is, how does the transcriptome differ between these two different cell types, the ones that are functionally coupled and not? So this general platform we found is pretty specific for targeting just those nucleus accumbens neurons that we think are activated by the glutamate inputs. So first, just by immunofluorescence, we can show that transgenic animals expressing the CHR2 in these glutamate axons indeed have many more mCherry positive cells compared to wild type controls. And then if we go in and then intracellularly patch from these mCherry positive neurons, we can see that they also exhibit higher photocurrents in response to blue light pulses compared to mCherry negative cells in the same slice. So this indicates that our FLARE labeling is pretty specific because it's targeting neurons that are more strongly synaptically coupled to glutamate inputs compared to the non-mCherry negative cells. All right, so how do we couple this now to something about transcriptomics? So again, you've seen these sort of plots and technologies being showcased during the past few days. So first in the in vivo animal, we'll perform a FLARE recording. So in this case, I'm just recording neural activity for 10 minutes while stimulating glutamate inputs. And then the following day, I dissect the tissue in the nucleus accumbens, create a cell suspension, generate a 10x library, and then do the sequencing. And so this is what this data looks like of all the cells. So I am just one person. I'm nowhere near the capacity yet of what you guys are at the Allen. So these are the cells that I have. So I have about 10,000 cells that have been microdissected from adult brains. These are the broad cell types that you expect to see when you do process this region. And these are the sort of top 10 gene expressions that cluster nicely along these different cell types. And so first, we just want to do sort of a sanity check. Are we able to even detect these exogenously introduced FLARE transcripts in our data? And so this is what that looks like. So if you remember on the previous slide, the neuron clusters were right around here. And when we plot the expression of our FLARE components, so the TEV protease, the transcription factor that's tethered at the membrane, and then our report of neural activity, you can see that they're largely confined to the clusters that correspond to neurons. So this is a good sanity check to make sure that we're sort of detecting the FLARE components. And then so among of our cells, about a little over 50% of them are in these neuron clusters. Among that, 30% express the FLARE virus. And among our FLARE positive neurons, maybe 10% are activated during our neural activity recording. All right, so now we can just subcluster just the neurons. So this is the population that I happen to be interested in. And we see many of the same cell types that you expect to see given all the striatal data sets that are publicly available now. So several different D1 MSN subtypes, D2 interneurons, even the cholinergic neurons popped up, and some cortex neurons eked into my dissection. So here's what these sort of top marker genes look like in this data. So if we plot the FLARE components now, just across these 9 cell clusters, what I found was that just this first cluster seemed to be upregulated in this TRE-mCherry activation. And so if we calculate an enrichment score of mCherry positive cells in FLARE positive neurons, again, it's significantly enriched in this particular D1 MSN subtype. And this was kind of really remarkable to me because when we do the clustering analysis, we omit these genes. So the clustering is being driven entirely by just sort of the endogenous transcriptome of these neurons. And it just happens to be that these mCherry positive labeled cells are falling into this cluster. So we were really excited about this. Just to be absolutely certain of this result, what I did then was rather than clustering all the neurons, I thresholded the neurons that highly expressed the FLARE components and then reclustered those to try to get rid of sort of any effects being driven by differential FLARE expression. And so when we do that of just these neurons that definitely are virally expressing FLARE, we get these 3 sort of smaller subclusters here. And again, this FLARE mCherry is defined to this one cluster. And in this case, we find other significantly enriched genes such as Calb1, PTN, TAC1. And cluster 2 is FOXP2, another D1 MSN marker. And cluster 3 is PINK, which is, of course, the D2 marker. Importantly, control genes, so GAD2, which is a universal MSN marker, and the TTA and TEV of the other 2 FLARE components are not significantly differentially expressed across the clusters. All right, so this is sort of giving me confidence that there's something very unique and special about this particular subtype that is getting activated by these glutamate inputs. And the real question for me was then, okay, so I think there's something molecularly unique about them, but functionally, are they capable of driving behaviors? And so can activation of these sort of downstream nucleus accumbens neurons I've identified also drive avoidance behaviors in these animals? And so based on this sort of transcriptomic data that I've collected, the first thought was, okay, well, we happen to have Calb1 and TAC1 Cree mice in the lab. What would happen if I just directly stimulated those cells inside of the nucleus accumbens? And so maybe unsurprisingly, especially since TAC1 is also highly expressed in this other D1 cluster, when you stimulate TAC1 neurons in the nucleus accumbens, you drive a very strong preference behavior in these animals. So these animals really tend to prefer to stay on the side of a chamber where they're receiving this TAC1 stimulation. And when I target the Calb1 neurons, I was really unable to find a robust behavioral effect. So these mice were very jumpy, and yeah, they don't seem to have any behavioral preference for Calb1 stimulation. All right, but FLARE the beautiful thing about FLARE is that it gives us genetic access to these neurons, right? So not only can we imagine driving expression of a static fluorophore, but we can switch out our transcriptional reporter, and instead of just a TRE-mCherry, we can express a TRE-mCherry fused to an opsin via a P2A. So this allows us now to not only visualize our neurons as before, but we can actually reactivate them using now a different color of light, so orange light to activate this red-shifted opsin. And so when we do this, so first when we deliver blue light to just activate these glutamate axons, of course we're driving this sort of mild aversive behavior in these animals. And then if we go in and stimulate with orange light, this cell bodies that are directly coupled to this glutamate input, we can recapitulate this behavior now and also drive an avoidance behavior in these animals. And this is in contrast if we sort of randomly label nucleus accumbens neurons in wild-type animals that don't have the glutamate axons labeled with CHR2. And so this is that data summarized there. All right, so what I've sort of had the chance to share with you all today is the example of this next generation of FLARE that enables as little as 1 minute labeling of activated neural ensembles in vivo. And using this new tool, we've developed a platform to kind of do anterograde functional circuit mapping in vivo. And this allows us to have both high throughput, specificity, and genetic access to the downstream coupled neurons. And we use this technology to molecularly profile neurons in the nucleus accumbens that fire action potentials in response to glutamate input in vivo, and discovered what we think is a cell type of, or subtype of nucleus accumbens neurons that we can access using FLARE, and then drive avoidance behaviors when we restimulate them using optogenetics. Future and ongoing work includes sort of in situ confirmation and spatial mapping of these FLARE-labeled neurons in collaboration with Xiaowei Zhang using MERFISH. I'm very interested in tracing sort of the axonal patterns of these FLARE-labeled neurons throughout the brain. Of course, we're always interested in intersectional approaches to trying to label these TAC1, Calb1, PTN positive neurons, maybe using some of, we're eagerly waiting the striatal data set from you guys on cell types so we can sort of see if there's any marker or way to access neurons like these using a specific enhancer. And I've also collected a simultaneous data set where I've labeled neurons that are activated by dopamine axon stimulation rather than glutamate. And so now we're comparing the sort of transcriptomic results we have between these two data sets. And finally, my last slide, I just want to share that when you're a tool developer, you want to think about how can you share this tool with the broader scientific community. And so in this case, I think for FLARE to really be a robust, usable tool, we want to have a transgenic animal that we can share with the community. And so this is something that we are developing with Basilica and Tanya. So we've already sort of generated these constructs and gotten them injected at UW. And so this is a transgenic FLARE mouse in which it's gatable not only with blue light, but we've incorporated a genetically encodable luciferase onto this calmodulin TEV-P component. And so now, rather than having to deliver exogenous blue light into the brain, you can inject this substrate of this nanoluc luciferase into the animal, and then this can permeate across the blood-brain barrier, go throughout the whole brain, and still activate this LOVE domain in a calcium and drug-gated manner. So in cultured neurons, we show that these cells can detect both light and high neural activity, and also this substrate firmazine and high neural activity. In vivo in a mouse, when we have nanoluc expressed in the prefrontal cortex and are collecting bioluminescence through an optical fiber, injection of this substrate can cause this bioluminescence that peaks around 2 minutes after IP injection and then dissipates around after 15 minutes. And so we're very excited about the potential for this technology to create this sort of scalable biology that people can study across the brain. And so with that, I just want to thank my advisor, Alice Ting. Mateo Lopez in the lab evolved the protease that we're using in this project. Sharu, Maisie, and Masa in the Diceroth lab characterized the RCamp for the calcium imaging data I showed. Paul Horabelt in the Malenka lab performed the patching, and I just want to thank my funding sources. And I'm very excited to take any questions.
Any questions? Yeah, wait for a microphone to come to you. Down. Christoph has a question down in front.
Yeah, beautiful talk. So I have two questions. A, how sensitive is the method at the single neuron or at the neuronal level? I mean, can you distinguish just on-off, or are there 10 different states you can distinguish? And B, related to that, you mentioned action potential. Now, actually, I never saw an actual potential. Have you ever done recording? So when you say your methods can pick up 1 minute, as little as 1 minute activity, do you have any idea, is that 100 spikes? Is that 1,000 spikes? Is that 2 spikes?
Right, so I didn't have a chance to share this here. So again, we've done these sort of simultaneous recordings just while doing calcium imaging, but as you all know, it doesn't always correspond to a single spike. So in that case, it's at least linearly correlated to the amount of fluorescence that we get from a calcium reporter. But the challenge that we're trying to do now with another electrophysiologist at Stanford is doing exactly what you say. So being able to drive just single action potentials in cultured neurons and seeing what the minimum number of spikes is that it takes to get reporter expression. I kind of feel that it's going to be a threshold, all or nothing, especially because of the transcription factor we're using. So if we swap it out for something that in our hands is a little more linear, then we find that I think it's a little more gradients of turn on rather than just all or nothing. But, yeah.
So that was a great talk. Thanks a lot. One of the interesting general questions about these cell classes, right, are the sources of the transcriptomic variation within them, right? They're sort of these blobs. They don't, map to individual points. And a specific question is how much of that variation might be activity dependent, right? Sort of variations in activity state. And I wondered if you could actually use the tool you've developed to maybe try and study that, right? To introduce different levels of activity and then see how that actually changed transcriptomic state in a sort of structured way.
Yeah, that's a great idea, especially so when we do the analysis, relying on the data sets that many other people have collected, they've sort of asked, what are immediate genes that seem to be regulated by whether the animal experienced some really prominent behavior that should have driven neural activity. So we exclude all those sort of known activity-dependent genes from our clustering analysis, but you're right, if we have a very defined way of driving neural activity, we could maybe get a much better handle on the sort of complete set of genes that are upregulated by activity in vivo. It's a good idea.
I have a question related to what Christoph was asking, but on the opposite side. Your activity reporter is really a calcium reporter. So it doesn't have to be correlating with spiking activities. It could be any kind of a post-synaptic signaling pathways that activate calcium. I was wondering, even in your discovery of the D1 pathway was preferentially activated, then let's say the D2 pathway in nucleus accumbens, would that actually because of the post-synaptic signaling specificity of the D1 cells?
I see, right.
So question is, have you looked at other conditions that can, non-spiking conditions, but signaling conditions that can activate your reporter, any kind of a GPCR or metabotropic GPCR pathways, for example?
Right, so I'm trying to think. So in cultured neuron, for example, where we have very good control, so you can imagine in D1 or D2 just active, causing these D1R, D2R antagonists or agonists. So I haven't tested yet whether that drives reporter expression of FLARE, but exactly like I would expect in D1 neurons, if I just stimulate dopamine release, it should cause this calcium reporter. So what I have done in vivo is, again, this is sort of indirect, but I've stimulated the dopamine axons, which should activate those D1 GPCRs. And in that case, I can get higher mCherry expression when I do that stimulation and not. And when I look at the transcriptomic data, the mCherry falls nicely into both D1 clusters I get rather than just a single one. And so I think it's specific in that way. But it's a good point, and I think sort of is motivating us to look beyond just calcium sensors, but to try and think of ways to design voltage readouts, and also for people who are interested in GPR-specific readouts, so activation of D1, D2. We have a glutamate sensor right now too as well. So
I would say the calcium readout is still very important. It's just capturing maybe more than just a spiking event.
then spiking, right? Yeah, absolutely. It's a good point.
Beautiful work, and I was wondering if you can explain a little more about what you see as the structure of the connectivity into the nucleus accumbens. It seems like the control experiment you showed, in which you took the slices and optogenetically stimulated the incoming fibers from the MPFC, that experiment could have been done before FLARE in a way as to identify whether MPFC neurons were specifically synapsing onto D1 or D2 neurons. And so I wonder why you think previous experiments maybe had missed this really striking finding that the MPFC inputs are specific to D1 neurons, and where do you think the D2 neurons are getting their input, if not also from the same cortical sources?
Right. Yeah. So I don't necessarily think that they're, that it's a very defined black or white, like D1 versus D2. And one thing that I think is sort of unique about doing this in an intact animal rather than in a slice is that all that sort of basal neuromodulatory input is happening in vivo. So that's also going to modulate how likely a neuron is to spike calcium, right? So it's going to change its threshold of spiking. And so, these D2 neurons, right, are normally inhibited by the tonic dopamine that's happening. So that's one reason why I think, maybe already they're at a disadvantage for spiking in response to glutamate input. So I think that maybe is what's happening here. The thing that I think is interesting is that it doesn't seem to be all D1 neurons that are getting activated by the glutamate, right? It's a specific subset of them. And so I think that would have been very, very challenging to identify in slice using that, because when you identify a D1 neuron, you back apply dopamine and everything will just start firing.
So in a different behavioral context, you would hypothesize that maybe the D2 neurons would show up if they were basally more active in that.
Yeah, exactly, right. So honestly, that was the initial. That's exactly what I thought I would find. I thought I would find, okay, sure, the D2 neurons are the ones that are going to get labeled, and not the D1. And so I was sort of really surprised to see this, and it's something that we're just like really diving into now to sort of figure out what's going on. Yeah.
Very cool. Thanks.
Okay, thank you so much, Christina. Another round of applause.